So greetings to all. In this session, we will see about recombinant DNA technology to produce transgenic plants and microbes. So what is recombinant DNA? Recombinant DNA are combined DNA molecules of genetic material from different or multiple sources. Recombinant DNA technology is a process of creating recombinant DNA by combining DNA from different sources and allowing them to replicate and synthesize the product of the gene which is transferred from the DNA of one organism into the DNA of another organism. So it is defined as it is a recombinant DNA technology is an artificial procedure by which DNA from different species can be isolated, cut and spliced together as a new recombinant molecules and are then allowed to replicate in rapidly dividing cells. Recombinant DNA technology is also called as gene cloning and in general it is called as genetic engineering. So now we will see about the basics of gene cloning. First, what is cloning? Cloning is a method of copying or producing similar products or things. For example, this is a pen. It has a cap, a cover and a lid and ink, and this is used for writing. Combining all these the components and the function, it is called as pen. So when we have a multiple copy of the similar kind of pen, this is called as clones of this pen. Similarly, what is gene cloning? So a gene is a, for example, this is a DNA which has a part, this is a part of genomic DNA which represents three genes, gene 1, gene 2, gene 3. What is gene? Gene is a part of a genomic DNA which represents a particular protein. So the function, it will synthesize a protein. So what is gene cloning? We need to copy this gene so that it will synthesize the protein in multiple level. So to this, what we have to do, we have to copy this gene. For example, this gene 2, we are making many copies. How we can do this many copies? There is a technique called polymerase chain reaction. Polymerase chain reaction is a method widely used in molecular biology labs to rapidly make millions to billions of copies of specific region of DNA using DNA sample and other components like primers, nucleotides, enzymes like polymerase used in a test tube using a machine called thermal cycler. So now we have copy of this gene, but originally for gene cloning we need to make copies so that it can replicate and synthesize the particular protein. But this copy which is made by this polymerase chain reaction cannot synthesize the protein, because to synthesize the protein it needs additional compounds and it needs some environment where it can replicate and synthesize. So we need to do few more steps in the cloning. So this is gene 2 and this gene 2 needs an environment where it can replicate and synthesize the protein which is called as host. So the host can be a bacteria or any other living organisms like plant. So now this gene 2 needs to be transferred it into the bacteria or the host. So how we will transfer? To transfer, we need a vector or vehicle which is known as vector. So this is a vector. So we have to insert this gene 2 into this vector and this vector can be transferred into the host where it will replicate and it will synthesize the protein. So to synthesize the proteins, this gene 2, it also needs few more components like upstream elements, which is normally called as promoter, and downstream elements, and For replication, it needs the origin of replication, and so these are also present in this vector. And when we transfer it into the vector and transfer it to the bacteria, here it will synthesize the particular protein. So now we know gene to vector and host, how it is transferred. So there are few more questions. How this gene 2 is made ready to insert into this vector, because we have to do some more things to make it ready so that it can be inserted into the vector. So what are those steps, and how the vector is made ready to accept this insert, then how the insert is transferred, as how this vector is transferred into the host, then once after transformation, how we will make sure that the vector which has our gene of interest is present in the host. So these are all few more questions which we need to get answer to know about the gene cloning. So before that, as a component of gene cloning, now we know one is vector, another one is host. So we will see more components for gene cloning, and when we see about the next components, we will try to find the answer for these questions also. The first one is restriction enzymes. So this will answer the question for how the vector and the insert is made ready to get fused together. So restriction enzymes are restriction endonucleases or restrictase is an enzyme that cleaves DNA into fragments at or near specific recognition sites within molecules known as restriction sites. So for example, if this is a restriction enzyme, so this is a restriction site. So it will cleave here and here. So it will make a break like this, and similarly here also it will make a break like this. So this is from one genomic DNA, for example from the vector, and this is from the insert, and this part and this part are complementary to each other so that it can fuse together. But so there is before that we will see there is two different kind of restriction enzymes are used in cloning. One is sticky end restriction enzymes and another one is blend end restriction enzymes. So these are all, this will make a protruding ends which is called as sticky ends, and the blend end restriction enzymes will make a blend end. Okay. So here, so now we know the restriction enzymes will cleave the insert and the vector so that we can fuse together. To fuse together, we need one more component which we need like a gum to stick, which is called as ligase. It is also a specific type of enzyme that facilitates the joining of DNA strands together by catalyzing the formation of a phosphodiester bond. So this is the sticky end from one DNA backbone. This is the sticky end from the insert. So when we combine together and in the presence of ligase enzymes, it will attach together, combine together and form the recombinant DNA. So now we need to see about one more enzymes which is used in the cloning. It is called as modifying enzymes. So previously we saw about, when we saw about the restriction enzymes, we saw there is one more kind of enzyme which produce blend end restriction sites. So for example, like this. So when we have a blend and restricted sites, and when we add the ligase enzymes, there is a possibility of the vector itself getting ligated without the insert, because this is a end, this is the blend end, and it can bind together so that it will close, it will ligate together and our insert will not get attached. So to avoid this, we need to remove the phosphate group in the 5' ends so that the vector will not self-ligate. For that, we need another one enzyme called as alkaline phosphatase. Alkaline phosphatase is a modifying enzyme that catalyzes the removal of phosphate group from the 5' end of DNA strand. This enzyme is frequently used in DNA subcloning as DNA fragments that lack the 5' phosphate groups cannot self-ligate. Now, we already saw about the vector, but we will see about the property of this vector now. So previously I told about a vector refers to any piece of molecule that contains genetic material that can be replicated and expressed when transferred into another cell. So the vector should have origin of replication where the replication starts. So this is a sequence from where replication starts, and piece of DNA when linked to this sequence can be made to replicate within the host cells. So apart from origin of replication, it should have a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Normally, it is some antibiotic resistant genes. So then it should have multiple cloning sites. This is some restriction sites where we can cut and we can insert our transgene or gene of interest. So cloning sites are in order to link the alien DNA, the vector needs to have very few, preferably single recognition sites for the commonly used restriction enzymes. Some examples of vector are plasmid, cosmid, bacteriophage, phagemids, bacterial artificial chromosome, and yeast artificial chromosomes. So now to summarize this basic of gene cloning, so we need a transgene which we have to amplify the gene of interest. Then we have to cut this gene of interest using restriction enzymes so that it can be ligated to the vector. Similarly, we have to restrict the vector to accept the transgene, and then we have to ligate this uh, transgene and the vector using ligase to produce the recombinant DNA. Once the recombinant DNA is produced, it has to be transferred into a host cell, so where it can synthesize, replicate and synthesize the recombinant, our transgene. So the components of gene cloning are insert, vector, host bacteria cell, and restriction enzymes, modification enzymes, and ligase. So in the next, in this video, we have seen about the basics of gene cloning. In the next video, we will see more about the transgenic microbes and transgenic plants.